Lots of weird little crystals, if you did it. Gauge strip, no specific gravities, normal ranges for things for urine, and things that can make your urine abnormal. That thing. The difference between BMI and body fat. Make sure you know how the little machine works. Okay, so look at your handout. Your handout is, you gotta make sure you study your handout. And then this is the digestion. So, 1, 2, 3 were gastric digestion. What did we put in the bottom of each test tube?
Pepsin.
Nope.
Oh, albumin.
Albumin. And what is albumin?
Protein.
It's a protein. Okay, so we put albumin in each. What did we put in test tube 1?
Pepsin.
5 mils of
Pepsin.
pepsin. What is pepsin? Yeah, we did. Talked about it in lecture yesterday.
It's an enzyme.
It's an enzyme that does what?
Digestion.
What kind of digestion?
Protein.
Digest proteins. Pepsin is a protease secreted in the stomach. Right? Okay. Second test tube you put in 5 mils of hydrochloric acid. Where do we find hydrochloric acid? In the stomach. And then the test tube 3, we put 5 mils of hydrochloric acid and 5 mils of pepsin. Okay, we put it in the incubator for an hour. Then we added Biret's reagent. Biret's reagent reacts with digested proteins to give a pink color. Undigested proteins will be purple. Test tube 1, test tube 2, test tube 3. What color did we get for test tube 1? What did we get for test tube 2?
Purple.
What did we get for test tube 3? What does that mean? Did we digest in test tube 1? Yes or no?
Yes.
We digested in test tube 1.
No.
No.
No. How do you know we didn't?
Because it's not pink.
Because it's purple. Did we digest in test tube 2?
No.
Why not? Why didn't we digest in test tube 1? No, there's protein there. Remember, you put a scoop of albumin in the bottom. Okay, let's go to the obvious answer. Why do we have digestion in test tube number 3? What is it about having both of those together that allows for digestion? How? What does the hydrochloric acid do?
It denatures the proteins.
So that
The pepsin to bring it down.
so the pepsin can get in and break it down, right? Why can't the pepsin do anything here? Because it's all folded up. We can't break it apart. Does hydrochloric acid digest?
No.
No. It doesn't cut the bonds. It doesn't break the bonds. All it does is open it up and leaves it as a whole protein. So you gotta have both. You gotta unopen it. You gotta unfold it, denature it, so that the pepsin can get in there and do its job. Does that make sense? And you've gotta understand this for next week. Have to. 4, 5, 6. Now we're in the intestine. So we put 5 mils of what in each test tube? Starch. What is starch? Carbohydrate, right? Is it a monosaccharide, a disaccharide, or a polysaccharide? It's a chain, right? Chains of carbohydrates, chains of sugars. So, You incubated this for 30 minutes, then we added iodine. Iodine binds to whole starches to give a black color. Okay? What color was test tube 5? Kind of tan. What was, I'm sorry, what was 4? 4 was tan, 5 was black, 6 was tan. I forgot to add what we did here. What did we add to the test tube number 4? Okay, we added pancreatic powder. What did we add to test tube 5? What did we add to test tube 6? 5 mils of spit. Okay. Where did we not have digestion? Where do we have whole starches left? Where do we have whole starches left? 5. Iodine binds to whole starches to give a black color. If it's a whole starch, did it get digested? So where do we have whole starches left? Test tube 5. So did we digest here in 5? No. How come? Why not? How come we didn't digest in test tube 5? Because water isn't going to do squat, is it? Water doesn't digest. Does nothing. All we did was add water to our starch. I'm not going to cut it up. Pancreatin powder. Turned tan. Did we digest? Yes. Why? Okay, what's in the pancreas that did the digesting? Which one? Which enzyme
Amylase?
digests carbohydrates? Amylase.
Amylase.
Amylase. Amylase digests proteins. Remember, your pancreas makes all the enzymes necessary. Amylase comes from the pancreas, digests our carbohydrates. Carbohydrates. Okay? Why do we have digestion in number 6? Because obviously we did. It's tan.
Your spit had amylase.
Because you have salivary amylase, right? And y'all had very good, powerful salivary amylase because it did its job in 30 minutes. Just as good as the pancreas. Alright, intestinal digestion 7. We added 10 mils of litmus milk. Okay, what did we add to number 7? Pancreatic powder. What did we add to 8?
Water.
Alright, so what we're looking for here is acid. We're looking for the color, we're looking for acid production. And we said acid will be evident by a dark purple color. Test tube 7, test tube 8. So, dark purple, light purple. So what are we looking for? We got acid. Who's got acid? 7. Why? Where does the acid come from?
Stomach.
What did we say we find in milk?
Fat.
Okay.
Sugar.
Alright, so fats. Carbs. Proteins. Which of these biomolecules would potentially result in the production of acid because of digestion?
The carb.
The carbs.
So if I break this away and I have a monosaccharide,
Protein.
is that acidic? Is a monosaccharide acidic? Nope.
So the proteins.
Why?
Amino acids.
Amino what?
Acids.
What? Those amino acids get removed, and now they're free floating. And what do? Make it acidic. What about fats? What's a fat made of? Glycerol, backbone, and
Fatty acids.
oh, what did you say?
Fatty acids.
I'm sorry, I still didn't hear you. Oh, fatty acid tails, right? And what do we do to digest those? Remove the
Cholesterol backbone.
fatty acid tails. And now we've got more free acid that drops our pH to make the acid to be dark purple. That makes sense? That's it. I gotta get ready for the second.